Here we're going to look at 4 examples of types of inheritance that don't match normal Mendelian genetics. So we kind of call these non-Mendelian genetics. All right, so our first one is the example of incomplete dominance. So we take our P generation. We're going to use chickens as our example. And these, I think, are Andalusian chickens. Anyway, so if we have a true breeding or pure breeding black chicken and a pure breeding white chicken, and we cross them, we find that all of the F1 are heterozygous. So here, in the case of incomplete dominance, the heterozygote offspring actually is like a blending of the two parents. So if this followed normal Mendelian genetics with complete dominance, let's pretend black feathers were completely dominant, then all of the F1 would have black feathers. But that is not the case. Instead, they're all a mixture of the two. And if we cross two F1 heterozygote chickens with each other, what we find in that phenotypic ratio in the F2 generation is a 1 to 2 to 1. We get 1 black chicken, 2 like grayish blue chickens, and 1 white chicken. So this type of inheritance of incomplete dominance is where neither allele is completely dominant over the other, and the heterozygote is the result of like a blending of the two alleles. It's like an intermediate form of the two parents or of the two pure breeding or true bred lines. Now, this is similar but also different than codominance. It's similar because the F1 generation, the heterozygotes, do appear different than what you would expect under Mendelian. Again, if it was Mendelian genetics, you would expect the F1 chicken to have black feathers. But instead, in codominance, the F1 have both traits expressed. So here we see a chicken with both black and white feathers. And if you cross two heterozygotes, again, you see that 1 to 2 to 1 ratio with the heterozygotes expressing both of the alleles. Now, you can remember this because the prefix co means together, like co-worker, collaborate, co-captain, like they work together or they show up together. Okay, so let's go ahead and see an example. So here we have two generation setups with flowers. And what I want you to do is think to yourself, which one is incomplete dominant and which one is codominant? All right. So over here on the left, we have incomplete dominance. We can see how that F1 is like a blending of the two parents or of the two pure breeding genotypes. And on the right, we have codominant, where the heterozygote has both phenotypes expressed. Okay, so let's go ahead and see this example of codominance with another new idea called multiple alleles. So blood types in humans come in a couple different options. So we have blood type A, and what I want to point out is when we talk about traits are based on proteins that you make. So in people with the genes for blood type A, the protein they make are represented by these blue spikes on this blood cell. Versus a person with blood type O has a mutation where they don't make any of these proteins. They don't have any proteins on the surface of the red blood cells. So it turns out that having type O blood is actually like homozygous recessive. Both copies of their gene do not have the directions on how to make proteins, whereas type A blood is dominant over O. So a person with type A blood could be homozygous dominant for A, or they could be heterozygous. As long as they have one copy of that A allele, they will make the A proteins found on the surface of the red blood cell. Now, a person, though, who is type B has the DNA directions to make protein B on the surface of the red blood cells. But protein B or gene B is also dominant over type O. So as long as a person had one copy of that gene coding for the B protein, they would have type B blood. Now, here comes in the codominance aspect. if a person inherits one chromosome that says have type A and one chromosome that says type B, each chromosome codes for their own proteins. So the chromosome carrying the gene for A will code for A proteins, and the chromosome carrying the gene for B will code for B proteins. So both proteins show up together. This is an example of codominance, not incomplete dominance, because they're both expressed in the phenotype. So here, when we look at this blood type trait, it is different than what we saw with Mendel. In Mendel, luckily for him and him figuring out the patterns of inheritance and how traits were inherited, his traits with pea plants only came in two possible alleles, either purple or white flowers, yellow or green peas. But in reality, traits can have multiple alleles. Here with blood type, there's 3 alleles possible. But as a diploid organism, we only inherit 2. So multiple alleles means there could be more than 2 options out there. And it just depends on which 2 you get that will determine your phenotype. Now, our last example of non-Mendelian, just kidding. Here's a Punnett square to show blood type and how you would do a cross for it. So let's take a blood type A and a blood type B couple and cross them to see what are possible in their offspring. So let's pretend they're heterozygous for type A. So they have type A blood, but they carry the allele for type O. Same thing with our type B blood. Let's say they have the dominant B allele, but also the recessive O. So when we look at their first possibility in offspring, this child would exhibit codominance and have type AB blood. And then they could have a child with type A, a child with type B, or a child with type O. All 4 possibilities are possible between these 2 parents. So this DNA here would code for both A and B proteins, only A proteins, only B proteins, or no proteins. Okay, great job. And now our last term to use that falls under the non-Mendelian genetics category is polygenic inheritance. I want you to think about this word, poly. Poly means many, and genic is genes. So here we are looking at a single trait that is due to many genes. So here is a human karyotype, and let's say we have gene A, gene B, and gene C, all located at different loci throughout the chromosomes, throughout the genome. So we have multiple genes, or polygenic here. So remember, genes code for proteins. So proteins from gene A, proteins from gene B, and proteins from gene C are all going to work together to determine the single trait of skin color in humans. So proteins from multiple different genes all lead to the beautiful variety that we can see in human skin colors. And it all depends on which combinations of these alleles a person inherits that will determine their skin color. So here, if you have 2 parents that are heterozygous for both traits, heterozygous for all of the genes involved in skin color, this right here represents all of the possible combinations and gametes that this one parent could make. And then over here are all the possible combinations that this parent could make. So this large Punnett square is showing us all the possibilities in phenotype of their offspring. So they could have, I'm still writing. They could have an offspring right here with like a lot of melanin produced, and their very next child could be this child. Both of them are possible from these 2 parents. So polygenic inheritance is when many genes influence a single trait. And that's it. There are a few other non-Mendelian things like epistasis and stuff that I'm just not including in this video. But otherwise, great job, guys.